All right. Hello, welcome everybody. Welcome to the webinar. We are going to go ahead and get started. My name is Madison Mueller, and I will be moderating today's webinar. If you have questions during the webinar, please use the Q&A. My apologies. If you're watching on YouTube, you can use the live chat function or the Q&A box on Zoom. Our panelists will be monitoring the chat to answer your questions, and we will likely have time at the end to answer questions as well. And Zach, if you want to go ahead and share your screen. Awesome. So in this webinar, Allen Institute for Immunology researchers will provide an overview of the research conducted by the Allen Institute for Immunology and share how multiplexed, multimodal fixed single-cell RNA-seq assays can overcome challenges found in traditional single-cell transcriptomics, including scalability and rare cell type identification. Further, they will demonstrate how the application of this multiplex assay to deeply profile cellular populations in human bone marrow can empower unique insight into a continuous developmental trajectory of cells. So for today's webinar, you will hear from 3 team members of the Allen Institute for Immunology. Peter Skeen has more than 15 years of research experience studying mammalian genome regulation, focusing on transcriptomics and epigenomics in both academic and industry settings. His interest in gene regulation began during his undergraduate and master's degrees at the University of Cambridge. Peter received a PhD from the University of Edinburgh. Now, as the director of high-resolution translational immunology at the Allen Institute for Immunology, he and his colleagues will apply single-cell technologies to profile gene regulation pathways of the immune system to further understand disease states and identify potential points of therapeutic intervention. Marla Glass is a human B-cell immunology scientist with experience in high-dimensional single-cell profiling and primary immune cell ex vivo assays. Currently at the Allen Institute, Marla leverages innovative single-cell technologies and wet lab assays, deep immunology expertise, and leading-edge data analysis tools to lead translational research projects. She received a BS in Biological Sciences from the University of Connecticut in 2010, and completed her PhD in integrative genetics and genomics at the University of California, Davis in 2016, during which time she investigated the interactions of an oncogenetic telomere integrating avian virus with the host T-cell genomic DNA. Marla also completed her postdoctoral training at Stanford University, where she studied IL-10 expressing B cells in healthy donors and organ transplant recipients. Zach Thompson is a senior bioinformatic analyst at the Allen Institute for Immunology. Prior to joining the Institute, he spent nearly a decade working in neuroscience-focused labs, investigating everything from how methylation in the APOE gene influences Alzheimer's risk to congenital brain defects in children. This work led to generating one of the first single-cell datasets on the development of the human fetal cerebellum. Now at the Institute, his main focus has been on using single-cell assays such as T-seq to probe how the healthy immune system changes over our lifespans, as well as assisting in new single-cell technology development. All right, and as mentioned earlier, Pete, Marla, and Zach are all team members of the Allen Institute for Immunology. And the Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, neural dynamics, cell biology, and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across our institutes, our scientific process is focused on tackling complex, broad, hard problems in fundamental sciences. Our approach is guided by 3 big principles: big, team, and open science, the most relevant of which today is open science. Our work generates data, knowledge, and analysis tools, which we share publicly, like in the webinar today. Now I'm going to hand it over to Pete, who will start our webinar on multiplexed single-cell multiomics for immune cell profiling and differentiation.
Hi, thanks everybody for joining today, and thank you very much, Maddie, for the introduction. So here at the Immunology Institute, we think of the immune system as a rheostat. So most of us hope to live our lives within the normal bounds of the immune system. We get colds, we get better. But overall, unfortunately, sometimes we either have too much of the immune system or too little. In the cases where we have too little of the immune system, we end up having a hyperimmune state. And here we study early and late stage multiple myeloma and malignant melanoma. We can also end up with the cases where we have too much of the immune system or a hyperimmune state. And here the body attacks itself in the cases of rheumatoid arthritis, Crohn's disease, ulcerative colitis, and cytokine storm and SARS-CoV-2 infection. We exclusively follow the human immune system. At this stage, we're not doing any mouse preclinical studies. And we follow patients who are in a longitudinal setting. So we have healthy cohorts and we follow people over a number of years. We have patients who've been diagnosed at risk of certain immunological diseases. We follow them through their transition to disease and how they may respond to treatment. For all these patient samples, we run a deepening profiling cadre of assays, and this includes flow cytometry, single-cell RNA-seq, single-cell epigenetics, high-plex proteomics, and the integration of human metadata. And we integrate all of these through vertical integration, and with the aim being that we can uncover the molecular linkages between the cell signaling cascade, going from the cell surface membrane with signaling proteins through intracellular signaling, transcription factor binding, transcription, and translation of response proteins. But one of the challenges is how do you support this at scale, and certainly in terms of multi-year longitudinal studies. And before we talk about exactly how we do that, I want to give you an example of some of our data in multiple myeloma. Here we're following patients who've been going through their induction therapy, comparing their baseline and drug treatment. On the bottom left, you can see there with flow cytometry, where we can see dramatic changes in cell lineage maturation. And then in the molecular assays and single-cell ATAC-seq and single-cell RNA-seq, we can see huge changes in the molecular landscape, either at the chromatin or gene expression level. So this really allows us to understand the molecular changes undergoing the induction therapy here in these patients. But one of the challenges is how to support this at scale. And in 2019, we developed and rolled out our 10x genomics v3.1 single-cell RNA-seq assay using cell hashing, which is a multiplexing approach that allowed us to significantly overload on the 10x platform and allowed us to multiplex many patient samples together and also include a bridging sample across every single batch. And this was really important as it allowed us to measure the technical variance between the batches. So here, if we just look at the bridging sample over 7 different batches, you can see that we actually only have a 0.1% batch variance. So very, very high quality of data. And this allows us to really drill deeply down with high confidence at the molecular changes across many batches where we have patient samples spread across a longitudinal setting. It also allowed us to save a huge amount of money. We've actually saved approximately $4 million over a non-hashed approach if we had just used a standard 10x genomics approach. And we published this work with Pollock Genge as the lead author in 2020. We've actually now generated probably one of the world's largest databases of single-cell RNA-seq. This graph is actually out of date now, where we've generated about 40 million cells following these patient samples across a number of different disease settings. But we always want to strive to become bigger and better. And now we're switching gears to a new technology, which is based on probe pair chemistry, shown in the bottom left here, that allows us to really magnify the amount of data that we can get from single-cell RNA-seq and dramatically drop down the cost. Here, rather than using a reverse transcriptase approach, we use these probe pairs to bind to the RNA in situ inside the cells, and then we ultimately count the cells that come, count the probes per cell. And this, through a multiplexing approach, really drives down the cost from what used to be thousands of dollars per sample, now to hundreds of dollars per sample. And this will allow us to expand our impact across many more patients and many more different diseases in the future. If we actually have a look at some of our preliminary data using this new approach and understand how, the quality of, on this new chemistry from 10x Genomics, called the Plex chemistry. And here we took our bridging leukopak and 7 healthy PBMC samples that were previously run on the old v3.1 chemistry. We split each of these 8 samples into 2 different replicates. So we had technical replicates totaling 16 samples. Samples were individually fixed and the barcoded probes were hybridized. We then ran this on the 10x Genomics platform at multiple different loading concentrations, 100K, 200K, which is the maximum recommended load-in, and then overloaded at 400 and 600K. If we have a look at the technical reproducibility between each of the paired technical replicates, so A versus A prime or B versus B prime, across each of the different loading densities, we can see a very high technical concordance here, typically at 0.99 R squared correlation, to very high technical reproducibility. And if we compare to the old V3.1 chemistry, and the gray dotted line is the one-to-one line, you can see with the new thick chemistry, we have a higher gene detection efficiency than the V3.1. So we have very high technical concordance. It actually has to reduce experimental risk, and we can actually store the cross-linked cells for further investigation. And we also have a higher gene detection at a reduced cost. If we have a sort of drill down into these quality metrics a little bit further, and we look at the cells recovered across different loading densities, you can see we have a linear tracking of the cells recovered. And if we have a look at the reads per cell, the UMIs or genes per cell, you can see it's flat across the different loading densities. So even as we're overloading the system more than 3 times the maximum recommended, we're not seeing a reduction in our gene detection sensitivity. And now if we compare to the V3.1 chemistry shown here in purple, and if we just look on the group of bars on the right-hand side, you can see we have approximately a 2-fold higher gene detection efficiency, even at the same read depth. So we have consistent performance at different cell loads. We can recover 300,000 singlets from a single well, and we have approximately a 2-fold higher gene detection. If we now just compare 2 of these samples and look to see our sensitivity measuring differential gene expression, we're comparing here A versus B, and if we show them in UMAP representation here, and then if we count the number of DEGs we can detect, you can see that we have a higher sensitivity for DEG detection with this new chemistry. But one of the potential risks of using probe-based chemistry is there's a missing search space. In the V3.1 chemistry, that captures all poly-A based mRNAs. So that's about 36,000 mappable genes. But with this probe-based chemistry, there are actually only 18,000 targeted genes, simply based on the number of probes that are targeted by the chemistry. So there's genes that are common to the platform, shown in gray, and then there's genes which are only visible in the V3.1 chemistry based on poly-A capture. So the question is, well, what are we missing when we move to this probe-based chemistry? To do this, we took our data where we had run the V3.1 chemistry in the case of baseline versus drug treatment in multiple myeloma, which I showed earlier. And now if we have a look at the genes that we can just cover and ask whether they are in the shared space, the gray region, or the red space, the genes that are unique to only the V3.1. And you can see the vast majority of the genes are actually in the gray space, so this is common to the platform. Which means that even though we're only now looking at these 18,000 genes, this is the genes that are really of interest in human immunology. So how do we actually implement this at scale? We've now arrayed this on a 96-well plate format. So we have patient samples thawed in subpools of 15, and we have a bridging control that's common, but it now is included in each subpool. And then we've automated this from an end-to-end approach, from thaw to library preparation, using a variety of different automation platforms. If we just have a look at the quality that we get from 2 subpools here, and we're just showing here these first 2 subpools, and we just look at the bridging control in each of these subpools. So we have pool A bridging control and pool B bridging control, and look at the technical concordance between these. You can see everything sits very much on the one-to-one line. And actually, if we have a look at the cumulative distribution, 99% of genes have an absolute difference of less than 0.143. So the data quality and reproducibility between the subpools is insanely high. So just to summarize where we're going with this, the new chemistry offers resolvable multiplexing at reduced costs. The probe-based chemistry allows higher gene detection efficiency, approximately 2-fold higher at the same sequencing depth, as the V3.1. And it's very compatible with deep immune profiling across disease states, despite only having a limited platform, number of genes tested. And we've now implemented this scale of very high efficiency. And now we're looking to roll this out over a large scale human studies. And then we're excited to see the data and the translational impact we can have with this new chemistry. With that, I'll hand over to Marla, who's going to talk a little bit more about how we're applying these technologies.
Okay. Thanks for that introduction, Pete. Now that we have given you a sense of the scale of data that 10x Genomics Flex technology enables, I'm going to talk about some of the exciting human immunology and immune cell development insights that we gained from applying this technology to human tissue samples. This technology enabled us to deeply profile cellular populations in bone marrow and blood at the single cell level using a systems approach. Here at the Allen Institute for Immunology, we specialize in conducting human-focused immunology research through a big science approach. As a result, we often perform deep analyses of cells in the bloodstream of healthy individuals in order to gain a sort of snapshot of the global state of the immune system in the absence of clinical disease. Additionally, study of healthy human bone marrow provides us localized insight on the critical features of immune cell development, as well as the supporting environment for this development that contributes to a functioning and mature immune system. Apologies for the delay. Collectively, understanding features of cell subsets in human bone marrow and peripheral blood tissue offers critical insight into immunobiology and is a very important precursor to understanding dysregulation of this biology in disease states and aging. Importantly, the bone marrow is the source of billions of blood and immune cells every day. As you can see highlighted in this table, a few of those populations that are generated in bone marrow. In fact, approximately 500 billion new blood cells per day may emerge from the bone marrow. That's in addition to those cells already circulating throughout the body. It is invaluable to understand baseline cellular composition, states, processes, and heterogeneity within this tissue for healthy individuals in order to comprehensively assess the issues that arise in disease states. Emerging high-dimensional single-cell technologies, such as fixed RNA site-seq, enable assaying all cell types, even rare progenitor populations in bone marrow. Using site-seq, we can align cells into a continuous trajectory, providing insights into cellular differentiation and the heterogeneity that exists in a traditional cell gate. Early development of B cells is a really excellent example of this utility. Early B cells originate from hematopoietic stem cells, followed by a common lymphoid progenitor and pro and pre-B cell stages, and finally emerge from bone marrow as an immature or transitional B cell. Hematopoietic stem cell transplantation is an absolutely essential therapy for the treatment of hematological conditions. So a more thorough understanding of hematopoiesis could enable more tailored interventions. For the experiment that I'm going to show you today, we collected matched bone marrow and peripheral blood samples from 4 healthy donors. We applied fixed RNA site-seq with a custom immune cell antibody panel to these matched tissue samples, enabling simultaneous quantification of 150 cell surface molecules and 18,000 genes. Importantly, we also multiplexed our samples, allowing us to combine them into a single cell pool before loading 400,000 cells into each of the 8 wells on a 10x Genomics chip, which is a lot higher numbers than we're used to talking about being loaded onto these chips. Using this workflow, We were able to deeply profile over 1.5 million cells from these tissue samples. And again, that's on a single 10x genomics chip. This enabled granular profiling of even the rarest progenitor cells, as well as mature immune cell types. We also observed tissue-specific features for those immune cell populations detectable in both the peripheral blood and bone marrow. This immense site-seq dataset with single-cell resolution of human bone marrow allows us to fully appreciate the continuous model of B-cell development. The summary map, heat map to the right, highlights the phenotypic marker expression of pro and pre-B-cell checkpoints aligned to reported populations across human B-cell development through a trajectory analysis. Prior proteomic studies, such as this mass cytometry-based analysis, have outlined the immune cell progenitor states that ultimately feed into the generation of functional human B cells. However, our site-seq data further enables insight into gene expression as well as molecular pathway engagement across the B cell developmental trajectory, and now at a comparable scale to cytometry studies. Here, we reclustered a B-cell developmental trajectory and utilized both RNA and ADT markers to provide higher resolution cell type labels than the reference datasets. Early B cells originate from hematopoietic stem cells, shown in the top center plot on the slide, followed by a common lymphoid progenitor cell, shown in the plot just below it, and then into a number of progenitor, precursor, and immature populations, shown in the plots just to the right, before migrating out of the bone marrow as more mature cells, represented in the far right plots here. After using a number of RNA and surface protein markers, we were able to identify the expected progenitor states along the developmental trajectory within this data set. Here we can see the same UMAP now labeled with their cell type designations from previous RNA and ADT markers. We see this great developmental trajectory through here, from our stem cell populations through these progenitor states and into more functional, mature B cells. If we now color the cells by their tissue of origin here on the right side, we see that progenitor populations are exclusively from the bone marrow, with the exception of rare circulating stem cells that we also captured. As we transition into these more mature B cell populations that are present in both peripheral blood and the bone marrow, we see that there is some separation in the UMAP between similar cell types, suggesting some molecular differences. We wanted to investigate this a little further by looking at the transitional B cell population, which is the earliest cluster in development where we see a mixed population of circulating bone marrow and peripheral blood cells. If we zoom in on these cells and separate them by their tissue type, we see that bone marrow B cells occupy the full expanse of this population, while the circulating transitional B cells fall further along this developmental trajectory, more towards the end of this particular cluster. By running a differential gene analysis test comparing the bone marrow and peripheral blood within this cluster, we find a number of genes, such as SLCA3 or GLUT3, CD83, MARCH1, and CXCR5, to name a few, that seem to reflect an increased capacity for engagement in both activation and functionality in peripheral blood, reflecting their further maturation as they've entered the bloodstream. Owing to the size of this single-cell data set, we are also able to gain insight into other rare B-cell subsets. One such rare and unique population is termed CD19 high, CD11C positive memory B cells, shown here, which are enriched in autoimmunity such as lupus and rheumatoid arthritis, infection, as well as in aging. We can also see that these cells can be separated by CD27 expression and that they exhibit potent immune signaling. If we zoom in on this effector B cell population, we can observe tissue-specific localization on this UMAP plot. In the lower right heat map, we've identified molecular features differentially expressed between bone marrow and peripheral blood effector cells. These differences could potentially show tissue-specific imprinting or differential migration patterns, to name a few hypotheses. If we then look at the differentially expressed proteins highlighted in this lower plot now, we see that these tissue-specific differences extend beyond molecular features for this effector population. For example, we observe that CD279, also known as PD1, the PD1 expressing subset rather, is unique to the peripheral blood population, to these effector cells. This type of large systems approach can generate hypotheses that we can take back to the bench for functional follow-up. So in summary, through deep profiling of human bone marrow and blood, we evaluated B cell developmental trajectories and mature B cell populations within the same individual. We found that transitional B cells in bone marrow and in peripheral blood exhibit unique molecular features. We also were able to profile the features of a very rare atypical B cell subset and found evidence of putative tissue-specific imprinting or differential migration patterns. In conclusion, this fixed RNA site-seq dataset enabled insight into components of a healthy immune cell state. And I'm now going to hand it off to my co-presenter, Zach.
Thanks, Marla. Let's see. Laser pointer. Cool. So today you've heard about how we're really trying to tackle profiling the immune system at scale. And part of that comes down to the data analysis and data management side of things. So I'm going to be wrapping up this webinar by going through our Human Immune System Explorer, also known internally as HICE. So HICE is our end-to-end research platform that allows us to gather, analyze, and interpret data. So we work with a number of external partners to collect both samples and clinical metadata that then get passed off to our wet lab team, who processes these samples through a number of single-cell technologies, such as single-cell RNA-seq, like you've seen today, as well as T-seq, flow cytometry, and O-link proteomics. This data can then be ingested into our HICE platform. And so for our single cell technologies, these actually go through an automated QC pipeline that I'll be detailing a bit more later on. We also ingest the clinical metadata collected by our external partners. And all of this can get fed into a scalable cloud-based analysis environment where our analysts can really tackle answering a lot of our experimental questions. Within this cloud-based environment, they can also build interactive visualization tools, and these can be shared with our scientists on site for collaboration, as well as creating publications that can be shared with the general public as well. So again, one of the key features of HICE is this automated QC pipeline and really managing this data at scale. And so here's just a little example of one of our QC pipelines and how it pertains to this particular experiment. So again, we had 4 healthy donors with matched PBMCs and bone marrow cells from each donor. And for the fixed RNA profiling kit, there's 16 available barcodes. And so for each sample, we split them out into technical replicates. So our wet lab team generated the libraries for this. And so they generated each barcoded sample and pulled into a single pool. This was then split over 8 wells of 10x and sequenced on a short read sequencer. Alignment for any 10x experiment is done on Cell Ranger. However, with 16 samples and 8 wells of 10x, we end up with 128 cell by gene matrices coming out from just a single batch. And so this is where the automated pipeline really kicks in. First thing is this automatic QC report that gets generated. This allows us to look for any sort of abnormal technical variation in a particular experiment, and we can drop any single sample, well, or batch that might have had some sort of issue in processing on the wet lab side. From there, we merge down all the wells by sample barcode, and then it goes through automated cell labeling as well as doublet detection. And so coming out of the pipeline, now we only have 8 H5 files with a single H5 file associated with each individual sample. And this really gives our analysts a head start on having cell labels already within here, doublets excluded, as well as really collapsing down a number of these cell by gene matrices. So this is obviously important within a single batch, but we've run over 150 batches to date through this particular pipeline. So this really helps us scale these technologies and really manage the data output from them. So one other aspect of HEISS that is really beneficial is the ability to generate these interactive visualizations and be able to publish them to the general public, and keeping with our mission of open science. And so at explore.allenimmunology.org, you can go visit this website, and there are a number of different publications from various scientists. And so I'll be giving a little demo of what one of those interactive visualizations look like as it pertains to this data set. Let's see if I can get this to play. So here's our public HEISS portal at explore.allenimmunology.org. And here we have a number of different publications from scientists at AFI. Right here we have our matched bone marrow and PBMC data set that we went over today. And within this publication, we have an accompanying Dash app visualization where you can actually interact with this particular data set. So within this Dash app, we can visualize by cell type. We can also look at a number of different metadata components. So we can look at tissue of origin. We can also look at donor effects and how these are distributed throughout this UMAP. We can also visualize both RNA and surface protein markers. So we have our global UMAP here. We can type in any of our favorite genes. So let's look at CD14, which is going to mark our CD14 monocytes. And again, we see this is very nicely specific to this particular cell subset. Besides the global view, we can also look at our B cell developmental trajectory. And again, here, when we look at cell type, we have our progenitor states, and then into these early B cell progenitors before they move into a transitional B cell, and into our more mature B cells in this bigger section. So again, on this UMAP, we can visualize both the transcriptome as well as surface protein. So if we look at the RNA MS4A1, we see this nicely highlights our more mature B cell population. While if we look at the surface protein CD10, we see that it will light up our more progenitor B cell types. So again, CD10 is really highlighting these more progenitor states, while it loses expression on our more mature B cells. So I highly encourage anyone to go check out explore.allenimmunology.org to take a look at any of the published data sets that we have on the website. So here's cool. And so with that, would like to thank everyone here at the Allen Institute for Immunology for all their hard work on projects like this and making this happen. A few individuals who I'd like to really highlight who contributed a lot to this particular data set. Aishwarya Chander has handled a lot of the data analysis for this particular data set, as well as Morgan Weiss, who generated all this data in the wet lab, as well as Claire for her guidance on the scientific questions. And so with that, we're happy to take any sort of questions.
Awesome. Thank you, Pete, Marla, and Zach. Just a reminder, you can use the Q&A box if you're watching on Zoom to ask questions, or the live chat option on YouTube. Our first question here is, how does the Allen Institute for Immunology plan on using this technology moving forward?
Yeah, that's a great question. I think we're really excited about the depth of the data and the number of cells we can study with this scRNA-seq chemistry. And I think we're looking to really extend the number of how we roll this out across a lot of different disease states and diverse human populations. So certainly stay tuned for that. We're also really excited about how we can add more multi-omic research as well. Previously, we had developed T-seq, which was a trimodal assay involving RNA protein epitopes and chromatin accessibility. And we'll be looking to see how we can do more multi-omic based research with this new plex chemistry.
Awesome. Thank you, Pete. Our next question are, what are the steps in the analysis of the B cells in this data set? Sorry, what are the next steps in this analysis?
Happy to jump in on that one. Yeah, it's a great question. So we will be moving in a number of directions with the follow-up B cell analyses, but really this has just been kind of a first pass analysis, a first glance at the data that I presented here today. But we have a lot of plans to look into some of the rare B cell subsets that in traditional scRNA-seq data sets you would have a hard time getting eyes on, just because of the rarity of those populations. So performing some further analyses on these rare subsets. One of those includes plasma cells, which are a really critical cell type for control of infection, and relevant in a number of diseases, even beyond infection. And so we'll be taking a look at some of the molecular and phenotypic features of plasma cells specifically, because of the depth of this data set. And furthermore, yeah, that's actually the bulk of our next steps. So yeah, great question. Thanks so much.
Great, thank you, Marla. Our next question is, what is the long-term vision for HEIS?
Yeah, I think I can jump in on that. So yeah, HEIS again is kind of our end-to-end platform, and we're really looking to expand sort of the public-facing side of it as we continue to publish a number of these longitudinal studies. And so we're looking to expand the amount of visualization tools and interactive ability with it, as well as potentially in the future having these as large healthy resources that people can integrate their own data or access to be able to look at how their own data compares with these published data sets.
Awesome, thank you, Zach. Our next question is, how do I learn more about our disease-based research?
Yeah, that's a great question. I mean, this talk, we're really talking, this webinar has mainly been focused on new technologies and about how we're rolling them out and how you can access that data through HEIS. I think, but fundamentally, our mission statement is to generate mechanistic insight into human disease transitions and the effects of therapies. So really, we're looking to roll out these technologies and our analysis into the disease states. You can definitely reach out to us through the website, and our email address is here. We can connect you with each of the leaders in our different disease areas. So that would be multiple myeloma, IBD and ulcerative colitis, SARS-CoV-2 infection, and our immune oncology program focused on multiple myeloma and malignant melanoma. So really happy to follow up and connect you with those right individuals. And please do click through our HEIS portal, and you'll be able to find a large number of our other studies.
Awesome. Well, I want to thank Zach, Pete, and Marla for their talks today, and I want to thank everyone for attending our webinar. As our presenter said, you can check out the Allen Institute website for more information on these resources, and we hope you all have a great day.